Welcome to NeuroLingo's second event. My name is Jeremy, and I'll be your host for this event. There are separate stream links for French and English, as shown in the description below. Feel free to switch to your preferred language at any time. Bienvenue au deuxième événement de NeuroLingo. Je m'appelle Jérémy et je serai votre hôte pour cet événement. Dans la description ci-dessous, vous pouvez trouver des liens différents pour visionner l'événement en français ou en anglais. N'hésitez pas à cliquer sur le lien pour choisir votre langue préférée à tout moment. NeuroLingo aims to make neuroscience research accessible to the public. The program is provided in the description. NeuroLingo vise à présenter les recherches en neurosciences d'une manière accessible au public. Le programme de l'événement est fourni dans la description de la vidéo. Today, students and researchers in neuroscience at McGill University will present their own research. Each talk is 10 minutes long, and we will have a Q&A period every 4 talks. Please use the chat to share your questions during the talk. Aujourd'hui, des étudiants et des chercheurs en neurosciences à l'Université McGill présenteront leurs recherches. Chaque exposé dure 10 minutes et une période de questions suivra chaque 4 exposés. Vous pouvez utiliser le chat pour partager vos questions durant les exposés. To open the event, Candice Cannone, a master's student, will present Von Economo neurons in depression and schizophrenia. Pour débuter l'événement, Candice Cannone, une étudiante à la maîtrise, présentera les neurones de Von Economo dans la dépression et la schizophrénie.
If you had to keep in mind only one positive aspect of a COVID-19 pandemic, what would it be? Some of you probably think, how dare she use the term positive and COVID-19 in the same sentence? And they're kind of right. But for me, it allowed us to put our attention on a very important topic, mental health. Did you know that mental disorders were one of the first leading causes of disability in the world? Because personally, before I started to work on the topic, I didn't. My name is Candice. I am a master's student at the Douglas Institute in the McGill Group for Society Studies. And in this talk, I want to present you the purpose of my researches, which consist in studying a particular type of neuron in 2 mental illnesses, depression and schizophrenia. But before to tell you more about what is this mysterious neuron I am interested in, I want to first take a moment to talk about mental disorders and show how important it is to investigate about them. Mental disorders have huge consequences on the daily activities and general behavior of the people they affect. It impacts their social, psychological, and economic life. In both depression and schizophrenia, there is a range of symptoms. And just to give you an idea, in depression, the symptoms consist in a mood disturbance that can be coupled with a lack of hope, of self-esteem, a deep feeling of sadness, and even suicide ideation. In schizophrenia, the symptoms can go from hallucination to a disorganized speech or behavior, some difficulties to concentrate, and there is even a syndrome called catatonia, which consists in a mental stupor and muscular rigidity. How to keep having a fulfilling life socially or professionally with all of those symptoms? 10% of the people suffering from schizophrenia die by suicide. And 50% of the individuals dying by suicide had previously received a diagnosis of depression. Mental disorders don't only reduce the quality of life of the individual they affect, but also their lifespan. Even if their perception in society is improving over the time, the patients suffering from schizophrenia and depression and all of the other mental disorders still to be victim of discrimination and stigmatization. And in the society, the severity of mental disorders tends to be considered as inferior in comparison to other illnesses. But the reality is that they all require an adequate medical assistance. And here is a sad example. 800,000. This is the number of people that died from a liver cancer in 2020. But this is also the number of people that died by suicide every year. Mental disorders are deadly illnesses. For those reasons, patients need treatments. And you already have treatments. But the thing is that a part of the patient doesn't respond to the treatment. And another part presents a resistance over the time. For those reasons, we need new treatments. But in order to find new treatments, we need to understand the mechanisms involved in those illnesses. And in the case of depression and schizophrenia, the famous neuron I study could be involved. This neuron, the von Economo neuron. What a fancy name! Von Economo. It comes from their founder, Konstantin von Economo, an Austrian neuroscientist that discovered them in 1929. Von Economo neurons also have nicknames. We can call them VENs as an abbreviation for von Economo neurons, or spindle neurons because they have a characteristic shape. In our brain, there is 100 billion of neurons that we can divide in 200 different types that have all their own features. And von Economo neurons are one of those types. Now, why I keep telling you this neuron is mysterious? Von Economo neurons haven't been studied so much in comparison to the other neurons. But from the few literature we have, we know that only a small number of species have von Economo neurons in their brain. And all of those species present some social and emotional abilities. We can cite raccoons, elephants, great apes, and humans. In humans, the von Economo neurons are not present in all the brain. They are specific to 4 regions only. And those 4 regions are involved in, again, social awareness and emotions. And we can cite social bonds, guilt, embarrassment, or again, empathy. And another interesting fact is that those functions are highly impacted in mental disorders, and that those same regions are also impacted in mental disorders, including depression and schizophrenia. Finally, the von Economo neurons naturally overexpress some specific genes that have been linked to depression and schizophrenia. So an alteration of the von Economo neurons could have a link with depression and schizophrenia. And this is exactly the question I want to answer at the end of my master. Is there an alteration of the von Economo neurons in depression and schizophrenia? I am interested in 2 types of alteration. An alteration of number, the density of the von Economo neurons, and an alteration of their shape. And those two alterations have a very huge importance, since they have the power to highly impact the circulation of the nervous message between the neurons. Now, to give you a visualization. Imagine that you're driving a car on a road. The roads are similar to neurons, and the cars are similar to the neuronal message. Now, the fact that the cars go from one point to reach a specific destination, this process is similar to the neurotransmission. And the von Economo neurons are kind of highways. They are way bigger than all of the other type of neurons, and they enable a fast neurotransmission. Now, let's imagine that the number of roads is reduced. It will be more complicated for a car to reach his destination. And if we imagine that now instead of having large and straight roads, we have some very small and sinuous roads, it will be complicated as well for cars to reach their destination. And this is exactly this way that the neurotransmission can be highly impacted if the morphology, the shape of neurons, or their density is impacted. In my researches, I compare the von Economo neurons from people suffering from schizophrenia or depression with the von Economo neurons from people that have healthy brain. How to do that? I work with post-mortem brain samples from donors. When people hear me talk about my job for the first time, tell me, "Oh my God, you cut brains. Isn't it disgusting?" But the reality is very far from zombie movies. The sample I work with are dissected by virtuoses of brain dissection. They separate the different structures of the brain, so the researchers only work on the sample they need to work with. When I receive my samples, they are tiny pieces of 1 centimeter length, like very little cubes, and there is no blood. Once I have my samples, I cut them in very thin slices, like 0.05 millimeters of thickness. To give you an idea, it looks like cherry blossom petals. Very far from the zombie movies, right? Then, when I have my little slices, I do a series of chemical reactions that will enable me to stain the von Economo neurons. And once this staining is done, I go to the microscope and I am able to see them. And here is an example at the end of the red arrows of the von Economo neurons seen under a microscope. Circled in green are the other neurons. And we can see that the difference between the von Economo neurons at the end of the red arrows and the other neurons circled in green is that the von Economo neurons are so big. We can also notice their particular spindle shape. So, thanks to the microscope, I will be able to determine the density of the von Economo neurons in healthy subjects in comparison to patients with depression or schizophrenia. And using 3D reconstruction, a specific software will help me to analyze every morphological features of the neurons and make comparisons between those different groups. This way, At the end of my master, I will know if von Economo neurons are disturbed in morphology or in number, in depression and schizophrenia in comparison to healthy subjects, depending on the region in which they are found. If I had to deliver only one message, it would be that mental disorders come from real physical brain disturbances, and that it is crucial today to keep finding new treatments and providing supports to the patients suffering from mental disorders. This is the aim of my researches and the heart of our scientific community. Thank you for your attention.
Thank you, Candice, for getting us started with this excellent talk. If you have any questions for Candice, please leave a comment in the chat, and we will address some of them in a Q&A session. Merci, Candice, d'avoir commencé notre événement par cette excellente présentation. Si vous avez des questions pour Candice, veuillez laisser un commentaire dans le chat et nous aborderons certaines durant la session de questions-réponses. Our next speaker is Shaima Alsuwaidi, a PhD student who will be presenting her project on using stem cells to study neurological abnormalities in immunodeficiency. Notre prochaine présentatrice est Shaima Alsuwaidi, une doctorante qui présentera son projet sur l'utilisation de cellules souches pour étudier les anomalies neurologiques de l'immunodéficience.
In the late 1900s, a one-day-old boy, who we can call John, was admitted into a hospital in Cleveland. John was born with a genetic disorder that caused him to have a severely compromised immune system. This disorder is famously known as bubble boy disease because patients were often kept in these protective plastic bubbles to prevent them from having deadly infections. Now, John stayed in the hospital for about 12 months before he was able to go home and continue to receive treatment. However, during his time in the hospital at around the age of 3 months, something interesting started to happen. John was having rapid tremors in his hands, as well as strange muscular movements in his posture when he would try to reach out for objects. And John was not the only one. More and more reports were coming out of patients with the same disease who were having similar symptoms such as tremors, learning delays, hearing loss, and more. Now, this is an interesting question, especially for me as a neuroscientist. How is this disease that is not neurological in nature causing these neurological symptoms? My name is Shaima, and I'm a PhD student working at the Douglas Mental Health University Institute. I'm studying one particular form of bubble boy disease, which is known as ADA-SCID for short, or as we scientists like to call it because we love to complicate things, adenosine deaminase deficient severe combined immunodeficiency. So the different types of SCID get their names depending on the genetic defect that caused them. And the particular type that I'm studying, which is ADA-SCID, is called that way because the affected gene is called ADA. And the question that my research focuses on is, how is the genetic defect of ADA-SCID causing these neurological symptoms in patients such as John? And basically, how is it affecting the nervous system? So now that we know what the question is, why isn't it easily studied, and how am I trying to study it? Well, it's not easily studied for two reasons. The first reason is that by the time that these neurological symptoms appear, the damage to the brain is already done, so to speak. And it's difficult for us to know exactly how it started. The other reason that makes it difficult to study this disease and other diseases that affect the brain is that we can't safely obtain a sample of patients' brain tissue because it's an invasive procedure, and I don't think anyone would want that done to them, and it can further damage the brain. So It's possible to do it in the case that a patient is already undergoing some kind of surgical procedure, but that's not always the case. So what do scientists do? Well, they either use animal models or post-mortem tissue, or they perform imaging scans on live patients to try and understand how a disease is affecting the brain. And while this has provided us with some information in terms of ADA-SCID, it doesn't allow us to understand how the disease is affecting the brain starting early in development, which is really important, and also how it affects the way that cells function. And this is where my research comes in to try and answer these questions. When I first became a neuroscientist, something that was really exciting for me was using stem cells in order to study diseases. So this is exactly what I'm doing. I'm using stem cells in order to study how the genetic defect of ADA-SCID affects the brains of patients and causes these symptoms. And this is a really exciting thing because even 50 years after the genetic cause of SCID has been discovered, we still don't have the answers as to how it affects the nervous system. So I hope that my research could try and answer that question. Now, what are stem cells? Well, you can think of stem cells as the baby cells that can then grow up to become any type of cell in the human body, such as your skin cells, your heart cells, brain cells, and much, much more. Literally every single cell that makes up your body. And these types of cells, like skin cells, for example, can be considered or thought of as mature cells. And in the human body, the cells would naturally follow the development from their baby state to their mature or grown-up state, just like us, basically. So how do these stem cells know what type of cell to become, given that they have so many options? They receive chemical messages which tell them what type of cell to become, and these messages depend on where the stem cell is located and also what surrounds it. And once the stem cell receives that message, it can basically switch off its genetic recipe for being a baby stem cell, and it can switch on its genetic recipe for being a specific mature type of cell. Now, the great thing about scientific research is that we have access to biobanks, which include biological samples from patients who have different types of diseases, even those who are unfortunately no longer with us, which is a great tool for us to use. So I obtain skin cells from ADA-SCID patients. And what I can do is I can use genetic engineering to essentially rewind them back to their baby state as stem cells. And this is really, really useful because now we can have access to stem cells from patients without necessarily getting them from an embryo. Now that I have these baby stem cells, I can then make any type of cell in the lab basically by mimicking the same chemical messages that stem cells receive in the human body that tell them to become specific types of mature cells. And one thing to keep in mind is that every single cell that we will then make will carry the same genetic defect that these stem cells from patients have. So I can specifically ask the question, how is this genetic defect of ADA-SCID affecting the way that brain cells develop and the way that they function? So to answer the question of how it affects the way they develop, what I make is an intermediate type of cell, which is called a neural precursor cell. And I can try and discover what kind of changes the disease is causing in these cells. And once I can discover that, I can then make specific types of mature brain cells, like neurons, glia, and other types of cells. And I can discover how these changes are affecting the way that they function. And one thing that you may have heard of before is that in scientific research, we need to have some sort of comparison between two groups for us to be able to make a meaningful conclusion. And in my specific case, the two groups are ADA-SCID patients and healthy individuals. And I'm lucky enough that through the same biobank, I can actually get skin samples from the healthy parents of these patients. And I can then perform all the same kinds of experiments on these cells to be able to make meaningful conclusions. So, to put things into perspective, we have this immune disease which is causing these neurological symptoms that have been a mystery for decades. And now, using the power of stem cells, we actually have the tools that we can use in the lab to try and discover what mechanisms are actually causing this. So as more and more discoveries are being made and advances are being made for treatments for the immune side of ADA-SCID, I hope that my research can also help us understand the neurological side of the disease so that we can also find treatments for those symptoms. And in doing that, patients like John would not only be able to survive outside of a plastic bubble, but like all of us, they can get a chance to thrive in all aspects of their lives. Thank you.
Thank you, Shaima, for your interesting talk. If you have any questions for Shaima, please leave a comment in the chat, and we will address some of them in a Q&A session. Merci Shaima pour cette présentation intéressante. Si vous avez des questions pour Shaima, laissez-nous un commentaire dans le chat et quelques-unes seront abordées pendant la session de questions-réponses. If you like the presentations, please give the streams a thumbs up. Si vous aimez les présentations, merci de laisser un j'aime sur la vidéo. Next up, please welcome Jason da Silva Castanheira, a PhD student who will be presenting his work on individual identification from brain activity. Veuillez accueillir Jason da Silva Castanheira, un doctorant qui présentera son travail sur l'identification individuelle à partir de l'activité cérébrale.
When you were a little kid, you were probably told to share your individuality with the world, express it from the top of the mountains. But what makes you unique? Maybe you like J-Lo? I absolutely adore Mariah Carey. Hi, I'm Jason. I'm a neuroscientist at the Montreal Neurological Institute at McGill University. And today I'd like to share with you a little bit about the research I'm doing throughout my PhD. Personality psychologists have tried to answer this question using personality questionnaires, finding traits that differentiate you from the crowd, like extroversion, how much you like to be around other people. Some individuals like to be surrounded by friends, be the life of the party, the center of attention, and other people prefer to be by themselves, alone in a park, reading a book. Whether you fall in one category or another, you're somewhere along the spectrum. But neuroscientists have had a much harder job trying to answer this question. How is individuality encoded in the brain? Historically, neuroscientists have tried to answer this question by taking brain scans of many individuals and averaging them all together to find the representative brain of someone who, for example, is extroverted. Finding brain regions that are more active in people who like to be around others in comparison to those who prefer to be by themselves. Historically, this has worked. It's allowed us to map brain regions that are more active in certain functions, allowing us to understand where certain functions lie in the brain. But there's one issue. It ignores a lot of information, because your brain scan is very different than my brain scan. So while we average information together, we're throwing away a bunch of individual differences. This is where my research comes in. I'm interested in these differences. We hypothesize that your everyday environment and everyday experiences shape the connections in your brain. They wire your brain. And so this, in turn, shapes your brain activity. And in essence, your brain activity should be very characteristic to yourself, kind of like a fingerprint. How do we record brain activity? We put individuals in a scanner, and we record the electrical and magnetic fluctuations of the scalp. As you may or may not know, brain activity is not static. It's constantly fluctuating up and down. Sometimes it's very slow, and sometimes it's very fast. And that's specifically what we're interested in, capturing these dynamics. The other thing we're interested in is looking at connectivity, how two brain regions communicate with one another. So with these two features, the logic is that if brain activity is characteristic to individuals, we should be able to pick you out of the crowd solely based on your brain activity. That's exactly what we did. With 158 individuals, we try to identify people solely based on the two features I described to you, the functional connectivity and the fluctuations in brain activity. And we achieve a great accuracy, above 90%. It's very impressive. So your brain activity is very characteristic to yourself. It's kind of like a fingerprint. Now, you may be worried. You may be thinking, am I next? Do the tech companies know about this? But you don't have to worry, because while the research is reliable and stable, the effects are not quite ready yet for commercial use. And I want to emphasize that the point of this research is to identify individual differences in brain activity rather than forensic identification of individuals. Next, we wanted to make sure that these results were stable. So we checked that it was reliable across days, weeks, and months. And we can reproduce this. The second thing is we looked at how long of a recording we need. We can do this with recordings as short as 30 seconds, which is very impressive. And lastly, we checked that it was something specific to brain activity and not something else you do in the scanner. Because you do all sorts of things in the scanner. You yawn, you stretch, you sneeze, you hum along to Mariah Carey. And so we wanted to make sure it's specific to brain activity. When we remove these artifacts from the recordings, we notice that it's actually brain activity that's driving the identification of individuals, which is amazing. Now, you might be thinking, oh, come on, I could have guessed that. My grandmother knows this. Of course, my brain activity is unique to myself. Duh. Well, I want to emphasize this is the first empirical evidence that brain activity is not only characteristic to individuals, but it's extremely stable. And so we can use this method in future research to try to help identify how differences are meaningful. That's exactly what researchers are doing around the world. People like Dr. Wheatley. Dr. Wheatley has taken brain scans of individuals and asked them to watch YouTube videos while they're having their brain scanned. It's not so hard. Then she asked people to rate the other participants as either friends, acquaintances, or strangers. What she finds is that two friends have very similar brain activity in comparison to strangers. So this, we think, is because friends share similar tastes, preferences, worldviews, environment, in comparison to a stranger where you might or might not have this. So it seems like brain activity seems to represent some sort of preferences or your individuality and is similar to your friends who you share certain features with. People like Dr. Kaufman have found that it might actually go wrong. You might be less identifiable under certain circumstances. Risk of mental health issues seems to be one of these circumstances where if you're at risk for mental health issues, you might actually become less identifiable across time. You're less like yourself. Your brain activity is less stable. Now, we don't know why this is, and we don't know what it means, but we hope that in the future, we can understand these things. And maybe this becomes a marker of mental health. So let's put it all together. What did we learn today? We learned that brain activity is characteristic to individuals. It's very specific and unique to yourself, kind of like a fingerprint. That these results are stable across days and weeks and even months. We can do this with recordings as short as 30 seconds, and these effects are meaningful. But this is just the tip of the iceberg. We hope that neuroscientists in the future can take this information and use it. Use this technique to help understand how individuals differ in terms of brain activity, how this might be a marker of mental health, and move away from averages where we lose all this information. And maybe, just maybe, we'll understand why you like J-Lo and I absolutely adore Mariah Carey. Thank you.
Thank you, Jason, for your enlightening talk. If you have any questions for Jason, please leave a comment in the chat, and we will address some of them in the Q&A session. Merci, Jason, pour votre exposé éclairant. Encore une fois, si vous avez des questions pour Jason, veuillez laisser un commentaire et nous aborderons certaines de ces questions lors de la session de questions-réponses. Upcoming is Gabriel Blanco Gomez, a master's student presenting about uncovering the genetic basis of language acquisition and neurodevelopment. À venir, la présentation de Gabriel Blanco Gomez, un étudiant à la maîtrise, une présentation portant sur le thème « La découverte des bases génétiques de l'acquisition du langage et du neurodéveloppement ».
Darwin's theory of evolution is fascinating, but there was one question that even Darwin himself found particularly puzzling. What is the origin of language? In his book, The Descent of Man, Darwin writes, language is one of the chief distinctions between man and lower mammals, but its origins are far more mysterious. How did we develop language? Is this something that's in our genes? I mean, if you think about it, babies do it effortlessly. By 4 months of age, a baby can already tell you the difference between vowels and random noise. We also know that language is universal. We can find it even in the most isolated places in the world. So it makes you question, is it something that we're wired to do? The same way that we're wired to walk or to blink? Is it something that's in our genes? If it is in our genes, can we find what those genes are? My name is Gabriel Blanco. I'm a neuroscientist, and this is a question that really interests me. How did language evolve? How did humans go and make that jump to have language? Well, when I first started, I thought the obvious place to look was primates. We share 98% of our DNA with chimps, which means that somewhere in that 2% should lie the answer. But if only science was that easy. That 2% represents millions and millions of combinations. So I had to change the strategy. And that's exactly what Dr. Fisher and his team did back in the late 90s. They realized that for us to figure out how something works, you have to look at examples and instances where it doesn't work. So at the time, they started to work with a family that had a particular genetic condition. In this family, almost every single member had some type of language impairment that went back generations. So what they did is decided to sequence that DNA and see if they could isolate the genes that were causing some of these problems. And then in April of 2001, they hit a major breakthrough. They found that a small mutation in a gene called FOXP2 was responsible for causing some of these language impairments in family members. A major scientific breakthrough. They had found the holy grail of evolutionary linguistics, the language gene, or as journalists called it, mankind's miracle mutation. Sounds cool, doesn't it? But remember how I told you science is never that easy? I'm going to ask you to put your scientist hat on and be skeptical, because it just sounds too good to be true. So when scientists took a closer look, they realized that there were some issues. First of all, FOXP2 is found everywhere in the body. We have it in our legs, in our lungs, in our heart. So how it relates to language, it's a little bit complicated. Second, we know that it's found across all species. Mice have it, as do dogs and elephants, giraffes, even our ancestors. A paper published in 2008 in Nature showed that Neanderthals had the exact same copy of FOXP2 that we do today, meaning that there was no evidence to suggest that there was some kind of positive selection. A kind reminder that in science, for something to be true, you have to be sure. You have to cross all your T's, dot all your I's, and make sure that it's perfect. So if FOXP2 is not the answer, where do we go next? I started at the beginning by saying that language is one of the chief distinctions of humans. But we also know that communication systems are found across all the animal kingdom. I mean, Elephants have certain calls, as do dolphins, dogs. There's even that weird, like, dozen or so people who genuinely believe their dogs can speak English, which, again, who am I to judge? Frankly, I actually like that because it really makes you think and reflect. What is really the difference between a dog and what we call human language? What makes our language so special? And the answer to that is that humans have this ability to take a finite number of sounds or vowels and basically combine them into an infinite number of sentences and words and phrases. Let me give you an example. Whereas a monkey can tell you when a tiger's coming, it's always limited by its particular context. Humans, on the other hand, we can go beyond. We can express complex ideas. We can tell you the tiger's coming, but we can also tell you about the weather. We can tell you how many stripes it has. Our language has something like a past, a present, and a future. We can tell you how we felt the last time we saw a tiger, how we'll feel in the future. Even better than that, we can talk about hypothetical scenarios of things we've never seen. We could tell you what we would do if we were to see, say, a blue tiger having lunch with Obama and Charles Darwin, because why not? Which, by the way, looks kind of something like this. And that ability to have or to communicate these complex thoughts, it's only possible if we use or if we have a complex machinery that allows us to do so. A software or a computer that is so complex and specialized that can process all kinds of information and make it into a way that we can be used to communicate with others. In other words, we need something like this. We need a brain. But then you might ask me, well, Gabriel, dogs also have brains, as do parrots and other animals. So what makes our brain so special? And to answer that, we're going to have to go back to 19th century France. In the night of 1855, a 50-year-old man walked into a hospital in Paris. Let's call him Mr. X. At first glance, Mr. X looked fine. He had no signs of stroke. He could walk. He could smile. But the one thing Mr. X couldn't do is he couldn't talk. Unfortunately, Mr. X will go on to die in the next couple of months. But his case wasn't unique. You see, at the time, there was a young neuroscientist by the name of Paul Broca who was carrying out autopsies on patients just like Mr. X. He really wanted to know what was going on in the brains of these patients that was preventing them from being able to talk or to speak. And then he realized that the one thing that Mr. X had in common with all these other patients is that he had a small lesion in an area on the left frontal lobe. He called it the language center. Another scientific breakthrough. Ask any neuroscientist today and they will tell you the same thing. Paul Broca's greatest contribution to science was being able to show that the brain is divided into different sections and that each section has a particular function. He also noticed something else, something weird. All of these patients had damage only to the left side of the brain. Why the left side? I mean, if you've ever seen a human brain, you notice that there are two hemispheres that look pretty much identical. So why is that language center only on the left? Why not the right? This is something that scientists or neurologists call brain lateralization, which is a complex term to say using one side more than the other. I'm going to ask you to put your scientist hat on again, because I'm going to ask you a question. And I want you to reflect. Think of your dog or the new puppy or kitten that you just bought. Are they left-handed, right-handed, left-pawed, ambidextrous? Reflect. If you're not sure, that's okay, you're not alone. Brain lateralization is very complex, and scientists are still not sure how it works. But what we do know is that it's very much present in humans. And not so much that. Our brains are wired to be lateralized, or to be right-handed. Something we have in our genes, if you will. Did you notice that pattern there? Genes, only something we find in humans, language. I think that would be a good time to do a little recap and try to see if we can put all of these things together. So what do we know? First, we can agree that language is something unique to humans, or at least our definition of language. 2, FOXP2 was not the answer, but it did teach us that if we want to find genes for language, we have to focus in the brain, because that's where we find most of the differences. And then 3, Mr. X's story was very tragic, but he showed us that humans have a language center, a highly specialized section in our brain only used for language. And that section is almost always on the left side of the brain. So putting all of these premises together, we can create some type of theory that goes like this. Genes lead to brain organization, and brain organization leads to language. I'll say it one more time because I think it's very important that you remember. There are a set of genes that tell our neurons to go to the left hemisphere, create that brain organization, and then that organization or that center is what eventually allows us to speak. But how do we find those genes? This is exactly why last year I decided to come to Montreal and work with children at risk for autism. Why autism, you may ask? First of all, nearly 50% of all autistic children develop some type of language disorder, with a large proportion of those being nonverbal. So autism and language are very close related. Second, whereas 1% of the population is thought to be ambidextrous, that number rises up to 35% in the autistic community. And no one really knows why. We think it might be something related to lateralization, but we don't really know. And lastly, we know quite a bit about the genetics of autism. So as a geneticist, if you want to isolate genes, it's better to work with things that we've done in the past. So I guess now would be a good time to tell you what I actually do every day, which I think is really cool. So in the lab, we bring these cute little babies. We put on an EEG cap. We collect their brain activity using electroencephalograms. And we have them watch a bunch of videos to see which side of the brain is more activated during which task. Little disclaimer here, we don't actually kidnap babies. We have their parents' permission, and they help us through the entire process. And what we've found so far, it's remarkable. Remember how I told you that language is found generally in your left hemisphere? Babies as young as 6 months old already have that type of lateralization and that language center. Babies at risk for autism, on the other hand, they seem to show no dominance between the left or the right hemisphere. What's even more interesting is that babies that have less lateralization go on to develop worse language outcomes. But what about the genes? I set out on a mission with one goal and one goal only, really. I wanted to find the language gene, that miracle mutation. And now it's time for me to tell you that, unfortunately, we haven't found it yet. We've come close, but we're not sure what that language gene is. But I think it's okay. Working with infants at risk for autism has taught me that our jobs as researchers is not only to make scientific discoveries, but to figure out how to use the scientific discoveries to change the world, to create better treatments, to help people. Isolating genes for language and measuring things like lateralization can help us diagnose infants earlier, which means we can have more personalized treatments. We can create better outcomes, help them communicate better, give them resources. So, no, we haven't answered Darwin's question. We've done quite a bit in the meantime. But I think that's okay. I mean, in a way, I think that's kind of the beauty of science, right? We start with a question, and we work through it. And although we don't always find the answer, we usually make different discoveries on the way. Because at the end of the day, all it really takes is just one question, one idea to change the world. Thank you.
Thank you, Gabriel, for this fascinating talk. If you have any questions for Gabriel, please leave a comment in the chat, and we will address some of them in the Q&A session. Merci, Gabriel, pour cet exposé fascinant. Si vous avez des questions pour Gabriel, veuillez laisser un commentaire dans le chat, et nous aborderons certaines de ces questions sous peu. We will now move on to our first Q&A session of the event. Our first 4 speakers, Candice, Shaima, Jason, and Gabriel, will answer selected questions from the chat. The speakers will address the remaining questions directly in the chat. Notre première session de questions-réponses de cet événement va maintenant débuter. Nos 4 premiers conférenciers, Candice, Shaima, Jason et Gabriel, répondront à des questions sélectionnées de vos commentaires. Les conférenciers répondront aux autres questions dans le chat. Please welcome Candice, who presented on von Economo neurons in depression and schizophrenia. Veuillez accueillir Candice, qui nous a fait une présentation sur les neurones de von Economo dans la dépression et la schizophrénie. Hello.
Hello.
So for our first question, I'd like to ask you, oh, sorry.
Oh, okay, I don't know what happened. My video just had been cut. I don't know why. Okay, I'm really sorry.
No problem. I can still ask the question.
Okay, it has to happen during a presentation. Okay, I'm listening to the first question, and I'm really sorry for this technical problem.
No problem at all. So for our first question, what kind of stain do you use to distinguish von Economo neurons from other neurons? Pour notre première question, quel type de coloration utilisez-vous pour distinguer les neurones de von Economo des autres neurones?
Alors, j'utilise, I use a staining and a technique called immunohistochemistry that use a specific antibody against a protein that is almost only expressed by the von Economo neurons. So once this process with antibody actually find the protein specific to the von Economo neurons, there is a series of chemical reaction with a substance called DAB that enable to stain the von Economo neurons. And then I perform a counter staining called cressyl violet, which enable to stain the other types of neurons. So I can see the different types of neurons in blue purple and my von Economo neurons in a gray black. So do you want me, I can answer in French if you want as well.
Yes, please do. Thank you.
Donc merci d'avoir posé cette question. Donc dans mon marquage, j'utilise un procédé qui s'appelle l'immunohistochimie, qui permet en définitive d'utiliser des anticorps qui vont reconnaître spécifiquement une protéine qui est caractéristique des neurones von Economo et qui, suite à des réactions chimiques, vont permettre, grâce à une molécule appelée le DAB, de colorer spécifiquement les neurones von Economo en gris-noir. Et donc ensuite, j'effectue un contre-marquage avec une substance qui s'appelle le cressyl violet, qui va permettre de marquer tous les autres neurones afin de voir les différents types de neurones qu'il va y avoir dans mon tissu. Et donc, voilà, je vais pouvoir voir tous les neurones en violet et les neurones von Economo se démarquer de par leur coloration gris-noir.
Thank you for that first answer. Merci pour cette première réponse. Pour la deuxième question, do you think that von Economo neurons could have important effects in other mental diseases? What other diseases do you think they might play a role in? Pensez-vous que les neurones de von Economo ont des effets importants dans les autres maladies mentales? Dans quelle autre maladie pensez-vous qu'elles pourraient jouer un rôle?
So the von Economo neurons haven't been that much studied in comparison to the other type of neurons. So there is only a limited amount of literature about the von Economo neurons. But among all of the literature, we have a lot of papers point out a link between a range of mental disorders and the von Economo neurons, not only with depression and schizophrenia, but also with suicide and psychosis, Alzheimer's disease, autism, or again, frontotemporal behavioral dementia. Donc, nous avons seulement que très peu de littérature au sujet des neurones von Economo, mais du peu de littérature que nous avons, une grosse proportion parmi ces articles concerne un lien entre les neurones von Economo et plusieurs maladies mentales, non seulement la dépression et la schizophrénie, mais également des maladies mentales ou des maladies cérébrales, telles que la maladie d'Alzheimer, l'autisme, la démence comportementale frontotemporale, le suicide en lien avec la psychose et l'abus de substances également, par exemple.
And for our last question, pour notre dernière question, can we become immune to mental disorders? Pouvons-nous devenir immunisés contre les maladies mentales?
Alors, la spécificité des maladies mentales, c'est que elles sont causées toutes, donc elles ont toutes des origines différentes, mais de manière générale, ce sont des maladies qui sont liées à des perturbations dans les mécanismes cérébraux. Et ce sont des maladies qui sont causées généralement de par des facteurs environnementaux et une génétique et une épigénétique, donc qui est le lien entre la génétique et l'environnement. Ce qui fait que ces maladies-là ne sont pas causées par un agent pathogène extérieur. Donc, il n'y a pas moyen d'être immunisé contre ces maladies-là du fait de leur origine. So what I was explaining is that mental disorders have results in a perturbation of brain mechanism that differs depending on the type of mental disorders, and the results in a mix between the genetic of the person, the environment, and what is called the epigenetic, so the impact of the environment on our genetic. And so they are not caused by an exterior pathogen agent. So our immune system cannot do anything against mental disorders. So there is no way we can be immunized against mental disorders.
Thank you for your answers. Merci pour vos réponses. So the remaining questions will be answered directly in the chat, and we will now move on to our next speaker. Les questions restantes seront répondues directement dans les commentaires. Nous allons maintenant passer à notre prochaine conférencière. Please welcome Shaima, who presented on using stem cells to study neurological abnormalities in immunodeficiency. Veuillez accueillir Shaima, dont la présentation a porté sur l'utilisation de cellules souches pour étudier les anomalies neurologiques de l'immunodéficience. Hello.
Hi.
So for our first question, pour notre première question, are these neurological issues from the immune system or are they caused by a different mechanism? Ces problèmes neurologiques sont-ils causés par des problèmes du système immunitaire ou par un mécanisme différent?
Thank you for the question. So what we're trying to identify with the research is whether the defect is directly caused or the problems are directly caused by the genetic defect. And the research that we have so far shows that this is the case, that the genetic defect of ADA SCID is affecting brain cells, so not just neurons, but also the immune cells of the brain, which are called glia. And there could be secondary results that are happening from the immune system defects, but the primary cause would be probably the genetic defect.
Thanks for that answer. Donc, pour résumer, actuellement, nous savons que les problèmes neurologiques sont très probablement directement causés par le défaut génétique à l'origine du DIX-ADA. Les problèmes immunitaires pourraient également avoir un effet supplémentaire. C'est pourquoi il est important d'étudier davantage comment le défaut génétique affecte les cellules du cerveau. So for our second question, pour notre deuxième question, do you think there is a possibility to predict an occurrence of a bubble boy before they are born? Est-ce que vous pensez qu'il serait possible de prédire qu'un enfant bébé ait un enfant bulle avant la naissance?
Great question. So basically, the way that ADA SCID occurs in children is something called autosomal recessive, which means that they need to be, both of their parents need to have the genetic defect, and they need to get a copy of each gene that is defective from both of their parents. So in that way, if the parents know that they have this genetic defect, then they can predict whether or not their child will get the disease. But other than that, they can perform newborn screening as soon as the child is born to see whether they have the deficiency or not.
Thanks for that again. Donc, pour résumer, cette maladie survient chez les enfants si les deux parents possèdent la mutation génétique. Si les parents savent qu'ils ont la mutation, ils pourront savoir si leur enfant peut contracter la maladie. S'ils ne savent pas s'ils ont cette mutation, ils peuvent également faire un dépistage néonatal dès la naissance du bébé et pour, dans ce cas-là, identifier la maladie. So, thank you, Shaima, again for your answers. Merci encore pour vos réponses, Shaima. The remaining questions will be answered directly in the chat, and we will now move on to our next speaker. Les questions restantes seront répondues directement dans le chat, et nous allons maintenant passer à notre prochain conférencier. Please welcome Jason, who presented on individual identification from brain activity. Veuillez accueillir Jason, qui nous a présenté sur l'identification individuelle à partir de l'activité cérébrale. Hi.
Hi.
So for our first question, Jason, pour notre première question, what does this mean for your personal data? Qu'est-ce que cela signifie au niveau des données personnelles?
That's a very interesting question. So like I mentioned, the point of this research is not forensic identification, but to understand the brain better and understand brain activity. We're not trying to identify strangers, and we're nowhere near that level where we can just take a brain scan and identify a stranger. So it's not going to be anytime soon where we're going to identify people kind of like you do on fingerprint sensors, like on your iPhone.
Thanks for that answer. Donc, pour résumer, comme je l'ai mentionné précédemment, le but de cette recherche n'est pas l'identification médico-légale, mais de mieux comprendre le cerveau et son activité. Nous sommes bien loin d'identifier des étrangers en utilisant une empreinte cérébrale, comme on l'utiliserait par exemple pour un iPhone. So for our second question, pour notre deuxième question, which scans are you using in your research? Are you combining several imaging techniques? Quels scans utilisez-vous pour votre recherche? Est-ce que vous combinez des techniques d'imagerie?
A great question. So I use magnetoencephalography, or MEG, which is an imaging technique that looks at the magnetic fluctuations of brain activity through the skull. Other people have used other imaging techniques like fMRI and EEG for what we call fingerprinting individuals based on brain activity, but my specific research is on magnetoencephalography.
Donc, pour résumer, excellente question. J'utilise la magnétoencéphalographie, qui est une technique d'imagerie qui capture les fluctuations magnétiques de l'activité cérébrale, mais cette technique a été appliquée à toute autre sorte de méthode de neuroimagerie, y compris l'IRM fonctionnel et l'électroencéphalogramme. So thank you again, Jason, for your answers. Merci, Jason, pour vos réponses. The remaining questions will be answered directly in the chat, and we will now move on to our next speaker. Les questions restantes seront répondues directement dans le chat. Nous allons maintenant passer à notre dernier conférencier pour cette première session de questions-réponses. Please welcome Gabriel, who presented on Uncovering the Genetic Basis of Language Acquisition and Neurodevelopment. Veuillez accueillir Gabriel, qui nous a parlé de la découverte des bases génétiques de l'acquisition du langage et du neurodéveloppement. Hi, Gabriel.
Hey there.
So for our first question, pour notre première question, is there any literature on FOXP2 function changing across the lifespan? Existe-t-il de l'information sur l'évolution de la fonction de FOXP2 au cours de la vie?
That's an interesting question. So we know that FOXP2 is very important for development. So it's highly expressed right before birth and in the first years of life, in the first 3 to 8 months, simply because it helps guide where the neurons are supposed to go. When it comes to the lifespan, so after those first years of life, research is not so clear. We know that sometimes it can be used as a transcription factor for different proteins and different contexts, but there's also some new research that shows that it's also expressed in the hippocampus. So we have an idea that it might be related to memory, per se, for example. Comme je dis, FOXP2, c'est tellement important pour les développements, le neurodéveloppement, surtout dans les premières 2-3 ans à la jeunesse en général. Mais après ça, comme les adolescents, même les adultes, nous, on n'est pas, c'est pas tout clair la fonction de FOXP2. On pense que c'est peut-être lié à la mémoire. On les trouve s'expriment, les gènes FOXP2 s'expriment beaucoup dans le hippocampus, qui est la partie, le centre de la mémoire. Mais la fonction en général, c'est un peu plus compliqué. Et ça veut dire qu'on a besoin plus des recherches, plus des études pour avoir une meilleure idée.
Thanks for that answer. For our second question, notre deuxième question, what do you think are the implications if when we find the language gene? Selon vous, quelles seront les implications si ou quand nous trouvons le gène du langage?
Okay, that's a really interesting question. I wish we were there yet. But one thing so first of all, it's when we talk about a language gene, it's not just a single gene. Like I think most likely we're talking about multiple genes. So that's something that I think it should be clear. Once we do find that multiple set of genes, the first thing that comes to mind would be, I think, just biomarkers for autism. Like if we know which genes are responsible for language, we can definitely help diagnose speech impairments or language disorders. We can help diagnose to see which kids are going to be verbal or nonverbal with autism. And I think that would be like the first main focus, like the clinical translation aspect of it. Now, if you want to go a little bit more into like science fiction or like what can we actually do, like we have included or injected genes for cancer in mice, for example. We can make models of autism in chimps or other different animals. So there's nothing really stopping us from putting those genes in animals to see if we can get some type of speech. Like I personally don't think it will be something like Planet of the Apes. Like we're not gonna have talking chimpanzees anytime soon. But it's one of those things that like we don't know. Like say we do find these language genes and we genetically modify some of these animals, what could possibly happen? It's something we don't know because we've never tried it. So oui, tout d'abord, si je veux dire que en ce qui concerne les gènes du langage, c'est pas qu'une seule gène, il faut noter que c'est plusieurs gènes. Et donc, à mon avis, je pense que la meilleure application, les choses qu'on peut faire, c'est si on sait que ce sont des gènes qui sont liés au langage, on peut les utiliser comme des biomarqueurs, c'est-à-dire qu'on peut prédire si les enfants auront des problèmes de troubles de langage, surtout dans les contextes de l'autisme, mais dans les autres problèmes de neurodéveloppement. Donc, pour moi, ce serait les bouts les plus importants, là qu'on trouve les gènes. Mais de l'autre côté, ça m'intéresse beaucoup parce que maintenant, dans la recherche, la science, nous sommes capables d'injecter des gènes sur des animaux comme des souris, par exemple. On peut les donner du cancer, on peut les donner l'autisme. Donc, ça veut dire qu'il n'y a rien de chose qui nous empêche vraiment d'injecter ces gènes du langage, entre guillemets, aux animaux. C'est-à-dire qu'il est possible qu'on pourrait avoir comme des animaux qui peuvent parler peut-être comme... Ça c'est un peu plus... ça va au-delà de mes attentes, mais on ne sait pas vraiment qu'est-ce qui va se passer si on essaie d'injecter ces gènes dans les animaux. Donc, à mon avis, c'est quelque chose qui... On n'est pas là, la science n'est pas là, la technologie n'est pas assez... On n'est pas dans ces moments, mais il y a plusieurs possibilités. Ouais.
So thank you again for your answers, Gabriel. Merci pour vos réponses. This is the end of the first Q&A session. The speakers will address the remaining questions in the chat. Ceci marque la fin de la première session de questions-réponses. Les conférenciers répondront aux questions restantes à travers les commentaires dans le chat. So our next talk will be given by Riya Thomas, a PhD student presenting her talk entitled Molecular Basis of Learning in the Cerebellum. Notre prochaine conférence sera donnée par Riya Thomas, une doctorante qui présentera un exposé intitulé La base moléculaire de l'apprentissage dans le cervelet.
Hi everyone. Pretty cool, right? No, not the glass, but the act of reaching out for a glass of water. I'm thinking a lot of you are still very confused. But do you know that this task is actually quite complex? This so-called simple task is so complex that scientists and engineers to date have not been able to engineer a robot that's as good at this task as we humans are. To put things a little bit more in perspective, let me narrate the story of my 10-month-old niece, Anya Baby. So, Anya Baby's favorite pastime is trying to throw her toys over the fence of her playpen. And one day, she decided to take on a task as huge as her. Literally, she tried to put the pillow over the fence of her playpen. The first few times she's trying it, she's not quite able to grasp the pillow properly. She keeps on trying and she's finally able to grasp the pillow and raise it some way, but loses her grip midway. Being the perseverant little munchkin that she is, she keeps on trying and finally she's successful at the task. The reason why I narrated this tiny anecdote to you is so that you can remember that at some point in all our childhoods, we have quite extensively trained so that we can make the task of reaching out for a glass of water so effortless. My name is Riya, and I'm really curious to understand how our brain is so good at performing fine motor movements and learning new motor skills. So the region of our brain that's concerned with these functions is called the cerebellum, and it's located somewhere in the behind part, and it's the region that's highlighted in red in the picture. And I'm interested in understanding how the cerebellum learns. Now, when you think of learning a new motor task, for example, throwing a basketball through the hoop, you know that the first few times you try this, you'll miss the basket or the target by a lot. But if you keep on trying, you get better with time. You might even get so good that you can make it into the NBA team. So how is it that we get better with time? The way our cerebellum does this is by actually getting feedback of our motor movement. So the feedback about the accuracy of your movement goes back to the cerebellum and is used to make your movement more smoother or accurate the next time. Before diving into details on what happens exactly in the cerebellum, let me remind you that neurons in our brain communicate through the language of electrical signals. So in the cerebellum, there's this interesting looking neuron called the Purkinje neuron. Pretty cool, right? It almost looks like how our hairs grew out during the pandemic. So this Purkinje neuron receives input from a group of cells called the granule cells, and it processes this information and sends it to other regions to finally generate a motor movement. Now, the function of the cerebellum here is to make your movement as smooth and accurate as possible. But when you start learning a motor task, the cerebellum is not very good at estimating how accurate or how good the movement would be. And you'll probably miss the target by a lot. Now, the feedback about the inaccuracy of your movement comes back to your cerebellum, back to your Purkinje neuron, in the form of electrical signals via a separate pathway called the climbing fiber pathway. This feedback is then used by the Purkinje neurons to alter the connections from the granule cells. So, if you remember, the information or electrical signals that comes in from the granule cells is actually what is used to control the accuracy or smoothness of your movement. So, refining or altering this connection would help to make that movement much smoother and more accurate. Now, if you can imagine, with trial by trial, your connections from the granule cells to the Purkinje neuron is getting more and more refined, so that the movement gets more and more accurate with trial, and you get better with time. Now, when you think of making that three-pointer shot, you might know that it's not just about getting the ball through the hoop. You're actually grasping the ball a particular way. You have to make a jump, then you have to actually throw the ball and get it through the hoop. Then you have to land back on your feet and not lose your balance. So there's a feedback that comes back to the cerebellum regarding each of these tasks. So what makes things a little bit more complicated is that all these feedbacks come back to your cerebellum, back to your Purkinje neuron, not at the same time, but at different delays. So for example, the feedback of whether you landed back on your feet and were able to maintain your balance comes in almost instantaneously, but the feedback of whether the ball went through the hoop comes in at a considerable delay. How is it that the same type of neurons, that's the Purkinje neuron, is able to process all these different feedbacks that's coming in at different delays? The way we think the cerebellum is able to deal with this is by having separate sets of Purkinje neurons assigned for each of these feedbacks that's coming in at different delays. So there's a set of Purkinje neurons for feedback regarding whether you were able to maintain your balance, and there's a separate set of Purkinje neuron for feedback regarding whether the ball actually went through the hoop. Now, what is interesting is that the only difference between these neurons, the Purkinje neurons, is actually in the expression of a few molecules. How is it that the difference in expression of a few molecules enable these Purkinje neurons to process information differently? This is exactly what our lab is interested in understanding. And we try to address this problem using the mouse brain cerebellum. So what I mean by molecules is actually intracellular components in the Purkinje neuron that is involved in receiving information in the form of electrical signals from the granule cells, processing this information through the chemical pathways, and then finally generating an output and transducing it back to electrical signals, which is the language of the neurons. So a peek into the work I do in my lab would look like taking mouse brain cerebellum, making live cerebellar slices. Then I take these live cerebellar slices under the microscope, and I'm actually able to view the Purkinje neuron. Then I approach the Purkinje neuron with an electrode so that I can record the electrical signals that is emitted by these Purkinje neurons, which is the information that the Purkinje neuron receives or transmits. And then to understand how the differences in molecules enable these Purkinje neurons to process information differentially, I block the function of these molecules and see how the processing of the information gets affected. Now, I think that trying to understand how our brain learns a new motor task or makes a fine motor movement is quite interesting. But this is not the only reason that we study the cerebellum. The cerebellum is also involved in a lot of neurological disorders. For example, ataxia, a lack of coordination in voluntary movements such as picking up an object or walking can be caused by defects in the cerebellum. Now, interestingly, so far I have talked to you about the role of the cerebellum in motor functions. But do you know that cerebellum is also involved in emotional and cognitive functions? Many studies point to the contribution or role of cerebellum in autism spectrum disorders. Now, to understand the cause and effect of malfunctions of the cerebellum in these disorders, we need to actually understand how the cerebellum functions. In our lab, for doing this, we go all the way from the behavior level down to the molecular level. Here, I'll conclude my talk, and I hope that the next time you reach out for a glass of water, you'll be able to smirk at those robots who have not got it right yet. Thank you.
Thank you, Riya, for this fascinating talk. If you have any questions for Riya, please leave a comment in the chat, and we will address some of them in the Q&A session. Merci Riya pour cet exposé fascinant. Si vous avez des questions pour Riya, veuillez les écrire dans le chat. Nous en aborderons certaines durant la prochaine session de questions-réponses. If you like the presentations, please give the stream a thumbs up. Si vous aimez les présentations, merci de laisser un j'aime sur la vidéo. Next up is postdoctoral researcher Marion Van Horn, sharing her research in wiring the brain with experiences. Ensuite, une chercheuse postdoctorale, Marion Van Horn, nous fait part de ses recherches sur comment configurer le cerveau à travers ses expériences.
The other day, I was working from home. I was sitting at the kitchen table at my office, and my son Ethan comes up to me and he says, "Mommy, one time I sat it too long and it hurted my leg." Well, I thought that was a pretty clever way of getting me up away from the computer, and I decided it was probably time that I put my work away and take him outside. But then I started thinking, maybe I shouldn't be taking him outside. Maybe I should be giving him an English lesson. He said, "Mommy, one time I sat it too long." You know, there's a few grammatical mistakes here. But then I also started thinking about all the time I'd spent with Ethan this last year. And I started thinking, what if I end up talking more like Ethan? Can you imagine? I'm on a Zoom call with a bunch of other neuroscientists, and here I am. Excuse me, one time I sat it too long and it hurted my leg. I think it's time we take a break and go outside. But I don't think you need to be a neuroscientist to know that no, even if you spend 450 straight days with a kid who hasn't quite mastered the English language, no, you will not end up speaking like him. But the question is why? Why is it that Ethan will end up talking more like me, and I'm not going to end up talking like Ethan? Well, it all comes back to the brain and how the brain develops. You've probably heard that the brain of a kid is like a sponge. A sponge, if you leave it on the table with some spilt water, the properties of the sponge are such that it will just soak up the surrounding water. And the same is really true for Ethan. I don't have to teach Ethan English. I don't have to teach him languages. If he's exposed to languages early on in development, he's just going to soak it up. Now, this is really because of our brain's ability to change in response to our environment. And this process of our brain changing in response to experiences is something known as neuroplasticity. Now, my interest in neuroplasticity goes above and beyond my 3 children. In a non-pandemic year, you would find me not only nurturing my 3 children, but you would find me in a research lab. My name is Marion Van Horn, and I'm a research associate at McGill University. I work in a research lab at the Montreal Neurological Institute, where I ask questions like, how? How does the brain change in response to experience? And the million-dollar question, what properties of the developing brain enable it to be so susceptible to change compared to the adult brain? And so I started by telling you that the brain is like a sponge. And I think that's a really good analogy, but I've looked at a lot of brains under the microscope, and I can confidently tell you that the brain is not a sponge. If you look under the microscope at a brain, what you'll see is the brain is filled with billions of nerve cells. Nerve cells, which we call neurons. So neurons are in our brain when we're born. We have billions of them. And these nerve cells, these neurons, they grow. They have branches, and they're actively growing. They're exploring. They're dynamic. And what these neurons are doing is they're looking to communicate their information with a neighboring neuron, with a partner. So they're looking for their partner to make a connection. And when they make this connection, which is known as a synapse, this is where they can transfer information. Now, early in development, when we're young, we have so much learning to do. And at this point in time, our brain is making tons and tons of synapses. We've seen that the brain goes kind of crazy and makes more synapses than it actually needs. Kind of like at the beginning of a pandemic, when everyone rushes out and buys way more toilet paper than they actually need. The brain is similar. It's making way more synapses, and eventually, there are just too many synapses. And so the brain has to decide which synapses to keep and which ones to eliminate. And we know through research that it's synapses, these connections that are stimulated, that get strengthened. And those that aren't used get pruned away, get eliminated. And so the brain is filled with these billions of neurons making connections, making synapses. But when I look under the microscope, there's more. There's more to the story. Our brain isn't just filled up with neurons, even though they've received a lot of attention. And we've done a lot of research trying to understand how neurons communicate. When I look under the microscope, and when others look under the microscope, we see more. We see that there are also glial cells, another type of cell in the brain that has received a little less attention. They were discovered over 100 years ago, and they were named glia. Glia in Greek means glue, and that's what we thought for many, many years. It's just that the glial cells were in the brain, holding the brain together, acting as support cells, the glue of the brain. Well, now our techniques have advanced, and we've been able to look at glial cells more closely. When we look at glial cells under the microscope and we study them, we see that they are also very active. They're dynamic. They have processes that are growing and exploring the environment too. They interact at synapses. They talk to neurons. And so they release substances like D-serine, which we have found is a very important molecule for strengthening synaptic connections. Researchers have also discovered that D-serine and other molecules that are released from glial cells can become dysregulated in certain neurological and neurodegenerative diseases like schizophrenia and depression and Alzheimer's disease. So we really need to keep studying these glial cells to figure out what molecules they're releasing and what role they play in learning and memory and neuroplasticity. There's another type of glial cell that we've started to study recently that's just really cool. They're called the microglia. And when we look at microglia under the microscope, what we see is these microglial cells are in the brain, and they're actively exploring, surveying the brain. And what we've seen is that these microglial cells will come in and they start nibbling at synapses. So we think that these microglial cells are really involved in the process of the elimination, getting rid of synapses that are no longer needed. And so that's really cool, right? That you have these cells that are getting rid of synapses. But I know what you're thinking. That's also kind of creepy. You've got these cells that are moving around in your brain, nibbling at synapses. Well, I try not to think about that too much. And so what this tells us is that we need to keep studying these cells. And recently, very recent studies have shown that these microglia cells, for example, are more active during sleep. And so we've known for many years, we know that sleep is really important for learning. We know that we need to get good sleep. If we're sleep deprived, we can't learn anything. And so we're starting to understand some of the processes that are involved in learning, and that when we sleep, these microglial cells are coming in and they're involved in eliminating synapses. And so we have a better idea that sleep is really important for the reorganization of the brain and setting these neural pathways. And so for me, as a mother of Ethan, what I know is not only does Ethan need to be exposed to a stimulating, waking environment, that I need to have active conversations with him and bring him outside so he sees stimulating sights and sounds, but also that I need to make sure that he's getting the recommended amount of sleep, because there's so much going on behind closed eyes. And so we have come a long way. Techniques and advances in science are happening every day. Researchers collectively are coming together and we're making discoveries and we're finding out new things about the brain every single day. And so what it tells us is we need to keep studying the brain because we're learning more. And what we know for certain is that the brain does change. The brain changes in response to our experiences and in response to our environment. And so if you do think back at your childhood, you can get a better understanding of how your brain was customized, and you can start asking questions about your brain. And if you do want to learn something, if you do want to change something about your behavior, know that neuroplasticity, this concept that your brain can change in response to your environment, it still happens in adulthood. It just takes a little more time and effort, and to be honest, it can be a little uncomfortable. But I have to tell you, I think this is a really good thing, because at the end of the day, we don't want to end up talking like Ethan. Thank you.
Thank you, Marion. If you have any questions for Marion, please leave a comment in the chat, and we will address some of them in the Q&A session. Merci, Marion. Si vous avez des questions pour Marion, veuillez nous faire part de ces questions dans le chat. Nous répondrons à certaines de ces questions lors de la prochaine session de questions-réponses. Our next speaker, Claudia Belliveau, a PhD student, will present on how child abuse impacts the brain of depressed suicides. Claudia Belliveau, une doctorante, expliquera comment la maltraitance des enfants a un impact sur le cerveau des victimes de suicides déprimés.
Child abuse is defined as physical, sexual, or emotional maltreatment or neglect of a youth, and it has devastating consequences on psychological development. Unfortunately, 1 in 3 Canadians has experienced some form of maltreatment before the age of 15. And we know that child abuse is a major predictor of depression and suicide later in life. Currently, 300 million people are suffering from major depressive disorder worldwide, and every 40 seconds, somebody dies by suicide. Interestingly, those who have major depressive disorder and were abused as children actually have more severe diagnoses of major depressive disorder. They have more severe symptoms, more recurrent episodes, and are usually less responsive to treatment. But the question is why? What is happening in the brain during child abuse that causes these negative consequences later in life? Well, we don't know everything that's going on, but today I'm going to explain to you some things that we do know. My name is Claudia, and I'm a PhD student studying neuroscience at McGill, and I study depression and suicide. So the first question is, what is normal development? Well, we know that childhood and adolescence are what we call a critical period of plasticity. During this time, the brain is very susceptible to things going on in our environment. It's kind of like a piece of Play-Doh. So the connections between cells are easily changed and rearranged, kind of like a piece of Play-Doh between your fingertips. And research actually shows that child abuse occurring during this critical period can alter the way that cells communicate, specifically in areas of the brain that are involved in regulation of emotion. So that kind of makes sense, right? Something severe happening during this critical period when the brain is very susceptible changes the way that the brain develops. The next question is, why is the brain of a child more susceptible than a brain of an adult? Well, in adolescence, our critical period of plasticity starts to slow down, and the brain goes from being a soft piece of Play-Doh to a hard piece of Play-Doh that can't really be changed. And how does this happen? Well, truthfully, there's a lot of things going on, but the most important to me is the development of these beautiful things I study called perineuronal nets, seen here. So these nets are made up of sugars and proteins that come together and form a literal mesh around certain neurons in the brain. And like us, our neurons need to be protected in a safe and enriching environment to grow to their full potential. And for us, usually our parents take on this role. They protect us from what's going on in the outside world, as well as from what's going on in our own mind. For the neurons, this role is taken on by the perineuronal nets. So these nets are actually there to protect the cells that they cover from the external environment of the brain, also from the toxic products that are produced when a cell is active. And unfortunately, we know that sometimes parents take advantage of their children and they abuse them. And we think that the abuse happening during this critical period of plasticity not only alters the way that neurons communicate, but also alters the way that these nets develop. So how do we study this? Well, I'm very privileged to work with the Douglas Bell Canada Brain Bank that houses over 3,000 brains of people who died from different disorders. And specifically, I look at the brains of people who died by suicide with or without a history of child abuse and compare them to a third group of psychiatrically healthy controls who died from reasons other than suicide with no history of depression. So I asked the bank for a piece of brain, and for me, that piece of brain comes from an area right above your eyes and in front of the middle of your brain, called the ventromedial prefrontal cortex. But don't worry, I won't quiz you on that later. What's important is that this region is responsible for regulating our emotions. So we stain the tissue so that I can see the perineural nets and the neurons. And I look at them under a microscope, take pictures of them, and then we individually count every perineural net. And we found that there is a major increase in these perineural nets in depressed suicides, but specifically in depressed suicides who have a history of child abuse. So we were right. Not only is the abuse affecting the neurons, but it's also affecting these perineural nets that are supposed to be protective. But what does this mean? Like, how is this happening? Well, we think that during the abuse, the children are developing maladaptive behaviors and coping mechanisms. And because of this increase in perineural nets, we see a solidifying of these maladaptive connections. And we think that this is bringing these maladaptive behaviors through adolescence and then into adulthood. And we think that this may be an explanation as to why suicide and depression are so highly correlated with child abuse. Unfortunately, due to the nature of my research, I can't know for certain that my hypothesis is correct because we are looking at only a snapshot of a person's life after they're passed away. But we think that we may have helped find a new target for antidepressant treatment, specifically for depression occurring after child abuse. Animal studies actually show that you can erase these perineural nets, and if you erase them in alive animals, you can actually erase some forms of memory, including fear memories. So my research is really important because not only can it influence public policy or help us understand what's going on in the brain of these kids that are being abused, but hopefully in 10 or 15 years, my research will have helped develop a new antidepressant treatment that can help lessen the burden of depression and suicide on the world and restore the protective nature of these beautiful things that I study, called perineuronal nets. Thank you.
Thank you, Claudia. If you have any questions for Claudia, please leave a comment in the chat and we'll address some of them in the Q&A session. Merci, Claudia. Si vous avez des questions pour Claudia, veuillez les écrire sous forme de commentaires dans le chat et nous en aborderons certaines lors de la session de questions-réponses qui suivra. Our final speaker is Yara Yakoub. She is a master's student whose research focuses on blood and imaging biomarkers in early Alzheimer's disease. Notre dernière présentatrice est Yara Yakoub, une étudiante à la maîtrise dont les recherches portent sur les biomarqueurs sanguins et d'imagerie dans la maladie d'Alzheimer précoce.
Every Saturday morning, beginning the summer of 2016, I would walk through this entrance of a local senior home into a grand lobby surrounded with a wall filled with composites of seniors' favorite photos. As I walk through the grand living room, I encounter Madame Wally, who I've been accustomed to meet every week swinging a baby doll on a crib. Every time I come in, she glance up, smile, and call me, "Hey, honey, come see your baby sister." For 4 years. Walking through the residence constantly sparked many questions, increasing my curiosity about the puzzle of the brain, all the structural, the functional changes that occur in the brain of a person developing Alzheimer's disease dementia. And I wonder if my fate would be one day similar to theirs as I grow older. Hi, my name is Yara Yakoub, and today I will be presenting part of what we do in the lab studying Alzheimer's disease dementia. On this day, after 15 months spent studying Alzheimer's disease for my research project, I recall Madame Wally's story, along with many stories that I heard of people living with Alzheimer's disease. And you may ask, we're in 2021. Don't we have a drug for this devastating disease? Well, my answer is not yet. However, the good news is that the research on Alzheimer's disease is on the march. Apparently, to find a drug that prevents Alzheimer's disease, we need to find ways to track individuals at the very early stages of the disease. Apparently, Alzheimer's disease pathology began to accumulate almost years prior to the cognitive symptoms become apparent. Proteins pile up to form sticky aggregates and twisted tangles between and within neurons or brain cells, ultimately leading to tissue loss and brain death. And this cascade began almost years, is known to be the preclinical or the pre-symptomatic stage of the disease. Currently, the diagnosis of Alzheimer's disease happens through clinical evaluation of the cognitive symptoms. However, this method is not a very effective tool because individuals come in with the late stage of the disease, where interventions may not be possible. So researchers try to develop brain imaging in order to track individuals at the very early stages where they are still cognitively healthy. PET scan is one example of such tool, in which a radiologist will come in, inject a small amount of a radiotracer material into the individual arm, and then this person would be put in the scanner. We will see if this tracer will bind to this protein pathology in the brain.
so that we infer that the pathology already started to accumulate in the brain of the person. While such tool is a very effective tool to track individuals at the very early stages of the disease, however, it's quite expensive. One brain scan can cost at least $2,000. Wow, that's actually expensive. I mean, at least for those who don't have publicly funded healthcare system like Canada. So researchers moved toward finding screening tools that are less expensive. And one such technique known to be the lumbar puncture or spinal tap. A specialist will come in, insert a needle into the spinal cord in between two bones of the individual, known to be the lumbar vertebrae. Will extract the cerebrospinal fluid collection, collect them, analyze them to identify the different protein pathology circulating in the body. While this technique is indeed more affordable method than conventional brain imaging, however, it might be invasive procedure, and not all individuals are comfortable doing such procedure. So what's the solution then? Imagine you walk into a blood lab in order to do a blood test to identify protein pathology in the blood. That seems something like science fiction or maybe impossible. But what if I told you if we are not very far away from this reality? Indeed, the recent development of different assay technology has made it possible to identify different proteins in the blood. Let me tell you a bit more of what we do in the lab studying Alzheimer's disease. We basically recruit individuals who are cognitively healthy, relatively younger, older adults group, age around 6 years old or older. They present to be cognitively healthy, no sign of memory impairment or any type of neurological disorder. However, one inclusion criteria for these individuals to be included is that they have a family history with the disease, putting them at a very high risk of developing Alzheimer's disease in the future. We take these participants, we do blood work, brain scans, and cognitive evaluation. And we wanted to know whether the blood work can actually reflect what we see of emerging pathology on the brain. And you know what? We were able to find that individuals with different levels of pathology in their blood actually also have an emerging pathology on the brain measured by PET scans. And not only this, participants with the very high levels of protein pathology in their blood indeed had worse, faster cognitive impairment over time. So that seemed like a very cool project, but what does it mean to others? Validating the plasma assays that analyze the different protein levels in the blood will help us implement such procedure, at least for now, in research settings, and perhaps one day in different clinical settings, so that such procedure will allow us to track individuals at the very early stages where they are still cognitively healthy, but perhaps have a high risk of developing Alzheimer's disease in the future. With the future of such possibilities, people like Madame Wali and others will be able to get ahead of the disease, and perhaps one day will be able to include such individuals in clinical trials, and perhaps one day we'll be able to find a drug that either stop or reverse the progression of Alzheimer's disease pathology, so that Madame Wali and others won't have to go through memory impairments and lose the capacity of identifying and recognizing others and their surroundings. Thank you.
Thank you, Yara. If you have any questions for Yara, please leave a comment and she will answer them directly in the chat. Merci, Yara. Si vous avez des questions pour Yara, veuillez laisser un commentaire et elle répondra directement dans le chat. We will now have our second Q&A session. Our last four speakers, Riya, Marion, Claudia, will answer selected questions from the chat. The speakers will address the remaining questions directly in the chat. Nous allons maintenant passer à notre seconde session de questions-réponses. Nos quatre derniers conférenciers, Riya, Marion, Claudia, répondront à des questions extraites du chat. Les conférenciers répondront aux questions directement dans la boîte de discussion s'il y en a plus après ça. So for our first Q&A, please welcome Riya. So Riya presented on molecular basis of learning in the cerebellum. Veuillez encore une fois accueillir Riya qui a fait une présentation sur la base moléculaire de l'apprentissage dans le cervelet. Hi. So for our first question, pour notre première question, can you directly apply the results observed in mouse cerebellum to humans? Pouvez-vous appliquer directement les résultats que vous avez observés dans le cervelet de souris à l'homme?
So I think there's a lot of similarities between the mouse brain cerebellum as well as the humans, say in terms of the type of neurons that we find or the connections between these neurons. And so whatever we find as like how the functions is affected in the cerebellum, we can apply or we can get an idea of how it happens in humans as well. But this still needs to be done with caution because there's a lot of difference between the two as well. So directly applying a result to humans won't probably work out. So we need to start with seeing whether the result holds true in research done on primates like monkeys, and then see if it holds true on human tissue that's obtained during medical procedures, and then like carefully translate it to how it will apply to humans.
Thanks for that answer. Donc, pour résumer, il existe plusieurs ressemblances entre le cervelet de la souris et celui de l'homme, par exemple les types de cellules ainsi que les connexions des circuits. Cependant, il existe toujours des différences entre les deux, et nous devons mener nos analyses attentivement afin de déterminer quels résultats peuvent être appliqués aux humains. Nous pouvons essayer de nous rapprocher davantage vers l'application des résultats au niveau de l'homme en vérifiant la validité de nos résultats dans les primates comme les singes. So for our second question, pour notre deuxième question, do you do tasks with mice to assess their motor function? Effectuez-vous des tâches avec les souris pour évaluer leur fonction motrice?
Yes, so we do this very cool task, which is called a porridge task with mice, where the mouse is actually made to reach out through a door, a tiny door, to reach for a food pellet or a like tiny bit of food. And why this is interesting is the instinct of the mouse is to actually obtain the food with its tongue. So it doesn't use its hand normally. And we are training it to use the hand to reach out and grab the pellet of food. And we can record these movements and see how fast they can learn this motor task, as well as track the movement of the arm to see how smooth the movement is. And then once we have this idea, we can see how these movements are affected when there's a malfunction of the cerebellum. So this behavior is something that's being done in our lab right now.
Thanks for that answer. Donc, pour résumer, oui, dans l'une des tâches qu'on effectue, qu'on appelle tâche d'atteinte des pattes, la souris est entraînée à saisir une petite boule de nourriture à travers une ouverture de 2 cm. L'instinct naturel de la souris est de ramasser la boule à l'aide de sa langue, et nous entraînons la souris à utiliser ses pattes au lieu. Et donc, ce qu'on peut faire, c'est on enregistre ses mouvements des bras et on observe la vitesse à laquelle elle apprend la tâche motrice et à quel point ces mouvements de bras sont fluides lorsqu'elle atteint la boule. On peut alors voir comment cette tâche est effectuée lorsqu'il y a des perturbations de la fonction du cervelet. So for our third question, pour notre troisième question, can the cerebellum be responsible for psychomotor delays in an adult? Est-ce que le cervelet est responsable des retards psychomoteurs chez un adulte?
So I think because the cerebellum is involved in motor function, and now evidences accumulate that it's involved in cognitive functions as well, deficits or malfunction of the cerebellum or lesions to the cerebellum that's caused even in adults could lead to deficits in motor skill acquisition. And this could, I think, manifest as delays in acquiring the motor skill or delays in acquiring the cognitive function as well, or learning a new task. But I would say that I would really need to look at elaborately at the literature of how it is actually linking to these deficits.
Thanks for that answer again. Donc, pour résumer, le cervelet est impliqué dans un retard de développement de l'acquisition de la motricité. Ces dysfonctions dans le cervelet peuvent provoquer des déficits ou des troubles de l'élocution, des difficultés à maintenir l'équilibre et d'autres déficits. Cela peut donc, à son tour, manifester par des retards dans l'exécution adéquate de fonctions motrices ou d'une fonction cognitive. So thank you again, Riya, for all of your answers. Merci pour vos réponses.
Thank you.
So the remaining questions will be answered directly in the chat. And we will now move on to our next speaker. Please welcome Marion, who presented on wiring the brain with experiences. Veuillez accueillir Marion, qui a discuté comment configurer le cerveau à travers d'expériences. Hello. Oh, sorry, I couldn't hear you there.
Sorry. Hi, bonjour.
Hi, very Montreal of you.
Yeah.
All right. So for our first question, pour notre première question, do you look at microglia cells in humans? Étudiez-vous les cellules de la microglie chez l'homme?
Yeah, that's a really good question and something that I didn't really go into much detail in the talk, but in general, a lot of these studies that we're doing are in animal models. So either in mice or in our case, all of our studies are done in tadpoles. And tadpoles are a really interesting model because we can study these tadpoles during development and they develop very quickly. So they're very small. We can look at them under the microscope and they develop very quickly. So we can study plasticity during development.
So thanks for that answer. Pour résumer, non, en fait, toutes les études sont généralement effectuées dans des modèles animaux. Par exemple, dans nos études, nous examinons un modèle de tétard. Ils sont vraiment cool parce qu'ils nous permettent de regarder ce qui nous intéresse et ils sont vraiment petits et se développent assez rapidement. So for our second question, pour notre deuxième question, what methods did you use to observe microglia activity during sleep in your lab? Quelle méthode utilisez-vous dans votre laboratoire pour observer l'activité de microglies pendant le sommeil?
Also, very good question. In general, we use, for all of our imaging, we do 2-photon imaging. So we look at these cells under a microscope where we can express different proteins that will basically glow depending on what wavelength we shine at them. But we haven't specifically looked at microglia in sleep in our lab, but it's definitely something that we're going to look at soon, hopefully very soon.
Thanks again for that answer. Donc, pour résumer, nous examinons ces cellules à l'aide de microscopie et nous utilisons la microscopie par excitation à 2 photons. En gros, nous exprimons quelque chose dans les cellules qui vont faire briller ces cellules lorsqu'on pointe un laser sur elles à une certaine longueur d'onde. Quelque chose à noter, c'est que le laboratoire n'a pas encore examiné la microglie pendant le sommeil, mais c'est quelque chose que nous sommes vraiment curieux d'observer bientôt. So for our last question, pour notre dernière troisième question, is it possible that microglia could eat up too many synapses? Are there diseases like that? And as a follow-up question, what happens if microglia do not eat up enough synapses? Donc, est-il possible que la microglie mange trop de synapses? Y a-t-il des maladies comme ça? Et comme suivi, que se passe-t-il lorsque la microglie consomme pas suffisamment de synapses?
Yeah, also very great question. So for sure, a lot of neurological diseases and neurodegenerative diseases are associated with issues with synapses. So for example, in Alzheimer's, you can have an issue where your synapses you're losing too many synapses. On the other hand, you have diseases or disorders like that are neurodevelopmental, such as autism, where we think that there are too many synapses. So when there are too many synapses, you can have connections that aren't necessary, and this can lead to sensory processing problems or things like that.
So thanks again. Donc, pour résumer, bonne question. Oui, c'est certainement une possibilité que cela puisse arriver. D'une part, dans les maladies comme l'Alzheimer, il y a une possibilité que les synapses soient perdues. D'autre part, dans les troubles de développement comme l'autisme, il se pourrait qu'il n'y ait pas assez de synapses. Donc, Thank you again for your answers.
Thank you
Merci
for
vos
helping
réponses.
translate.
Trying my best. All
Yeah.
right. So the remaining questions will be answered directly in the chat. And we will now move on to our next speaker. Les questions restantes seront répondues directement dans les commentaires du chat. Et nous allons maintenant passer à notre prochaine conférencière. Please welcome Claudia, who presented on child abuse impacts the brain in depressed suicides. Veuillez accueillir Claudia, qui a fait une présentation sur la maltraitance des enfants a un impact sur le cerveau de victimes de suicide dépressif. Hi.
Hi.
So for our first question, pour notre première question, how can you know about their abuse history if you're using brains from people who are deceased? Comment pouvez-vous connaître les antécédents d'abus de donneurs qui sont décédés?
So the Douglas Bell Canada Brain Bank actually has a team of professionals that this is actually their job. So we have professionals that work with us as well as psychologists that work with us. And so they perform something called a psychological autopsy. So what this means is that they get all of the clinical information available for the brain that we receive. So whether that be medical files, social work files, interviews with the families, interviews with the doctors that are on the cases, and we get information from the police as well. So basically, we take all of this information and create sort of like a characterized history of the brain that we receive, and we only include very severe abuse in our cohort, so in our study, to make sure that when we compare, to psychiatrically healthy controlled persons, like somebody who doesn't have depression or abuse in their history, like we really see both ends of the spectrum and we're not kind of getting lost in the middle.
Thanks for that answer. Donc, pour résumer encore une fois, la Banque de cerveau Douglas Bell Canada dispose d'une équipe de professionnels qui effectue ce que nous appelons une autopsie psychologique. Cela signifie que les antécédents médicaux sont transmis à notre équipe, puis des entretiens sont menés avec la famille et le médecin. Cela nous donne une description précise de l'enfance des donneurs et dans notre étude, nous incluons seulement aussi des cas d'abus graves. So for our second question, pour notre deuxième question, what's the process that leads to the brain? What's the process that leads to there being more perineuronal nets? Quel est le processus qui fait qu'il y a plus de filets périneuronaux?
So I think my internet connection is unstable, so I apologize. That's what my Zoom is telling me. So we actually don't know exactly why there are more perineuronal nets, but recent literature suggests that the helper cells that Marion was talking about, so the microglia, may actually be involved in regulating the net. So they may literally go up to the net and eat it away or secrete things, which means like put little chemicals out into the brain that can either support or again, eat up the nets. So yeah, I'm super excited. Some research that I didn't show to you today. I have been looking at microglia in my brain samples, and we do see that there is a dysregulation in these microglia that should be regulating the nets as well. So I think it has something to do with the microglia.
Thanks again. Donc pour résumer, en fait, nous ne savons pas encore exactement, mais les recherches récentes suggèrent qu'il y a des réseaux périneuronaux qui sont régulés par des cellules auxiliaires, comme la microglie que Marion a mentionnée aujourd'hui. Ainsi, il semble que la microglie ne dévore pas seulement les synapses, mais qu'il dévore également ses filets. Une partie des résultats de ma recherche montre en fait que les filets périneuronaux sont également régulés chez les patients, les personnes maltraitées. Il nous reste donc encore beaucoup à apprendre sur la façon dont la microglie fonctionne réellement pour réguler ces filets. So for our next question, and the last question, pour notre dernière question, do you think exercise can alter the perineural nets? Pensez-vous que l'exercice peut changer les filets périneuronaux?
So There's actually some studies that look at exercise in a rat model. So they have a rat that sort of exercises when they want and one that is not allowed to exercise. And there's some labs that look at perineuronal nets in the spinal cord as well, so not just the brain. And I know of one paper where they actually compare the number of perineural nets in the brain before and after exercise, as well as in the spinal cord before and after exercise. And there actually is quite drastic changes in the amounts of perineuronal nets. So we know, or research has showed us over the past 30 years that perineuronal nets are very stable, but actually we're learning more and more that they're quite dynamic. So they change when you sleep or they change when you exercise. So yeah, get your exercise in.
All right, make sure to. Donc, pour résumer, il existe en fait des études qui comparent l'expression des filets dans le cerveau ou dans la moelle épinière des rats qui font de l'exercice ou non. Ces études remarquent en fait que les différences dramatiques dans le nombre de filets, mais pas entièrement de la façon dont on imaginait. Nous savons que les filets périneuronaux sont stables au cours du temps, mais nous en apprenons plus, en plus qu'ils sont également très dynamiques et qu'ils sont affectés par les cycles de sommeil et ou l'exercice. So thank you again for all of your answers. That was great. Merci encore, Claudia, pour vos réponses. The remaining questions will be answered directly in the chat, and we will now move on to our next speaker. Les questions restantes seront répondues directement dans les commentaires du chat, et nous allons maintenant passer à notre prochaine conférencière. Please welcome Yara, who presented on blood and imaging biomarkers in Alzheimer's disease. Veuillez accueillir Yara, qui a présenté sur les biomarqueurs sanguins et d'imagerie dans la maladie d'Alzheimer précoce. Hi.
Hi, good evening.
Thanks for joining us. Donc, pour notre première question, for our first question, are there any factors that affect the levels of Alzheimer's disease protein pathologies in the blood? Existe-t-il des facteurs qui affectent les niveaux sanguins des protéines concernées dans la pathologie de la maladie d'Alzheimer?
That's a very interesting question. Indeed, yes, there are different modifiable and non-modifiable risk factors that can play a role in Alzheimer's disease pathology, and therefore the concentration of different pathological proteins in the blood. Some of the non-modifiable factors, for instance, age alone can play a very huge risk factor in the development of Alzheimer's disease, sex, and genetics, for instance, like ApoE, that's been seen to be the most a genetic risk factor in the sporadic non-genetic form of Alzheimer's disease. And the modifiable forms such as the modifiable factors like stroke, chronic kidney disease, and the body mass index in general, they all affect the concentration of these proteins in the blood.
Thanks for that. Donc, pour résumer, oui, en effet, il existe différents facteurs modifiables et non modifiables qui jouent un rôle dans la maladie d'Alzheimer et donc affectent les niveaux sanguins des protéines pathologiques. Les facteurs non modifiables tels que l'âge, le sexe et la génétique sont considérés comme des facteurs de risque à la maladie d'Alzheimer. Et ce sont donc des facteurs qui affectent les niveaux sanguins. Les facteurs modifiables tels que les accidents cérébrovasculaires et les maladies rénales chroniques ont été associés à des niveaux de protéines amyloïdes et tau, qui sont les deux principales caractéristiques pathologiques de la maladie d'Alzheimer dans le sang. So, thank you for that great answer, Yara. Merci pour votre réponse aujourd'hui. This is the end of the Q&A session.
Thank you.
The speakers will address the remaining questions in the chat. Ceci marque la fin de la session de questions-réponses. Les conférenciers répondront aux questions restantes dans la boîte de commentaires. So thank you again for joining NeuroLingo today. A shout out to the speakers for making this event an absolute blast. Special thanks to the speaker trainers, Dr. Edward Ruthazer, Dr. David Ragsdale, and Nicolas Belliveau for coaching the speakers for the past 4 months. Merci d'avoir rejoint NeuroLingo aujourd'hui. Un grand merci aux conférenciers pour avoir fait de cet événement un véritable succès. Et un merci particulier aux formateurs de conférenciers, le Dr. Edward Rutazer, le Dr. David Ragsdale et Nicolas Belliveau, qui ont encadré les conférenciers au cours des 4 derniers mois. Today's event was supported by the Tannenbaum Open Science Institute at the Montreal Neurological Institute, the Integrated Program in Neuroscience at McGill University, the Douglas Mental Health University Institute, the Graduate Students Association in Neuroscience, and the Postgraduate Students Society of McGill University. Finally, we would like to extend our gratitude to those of you who donated during the event registration. L'événement d'aujourd'hui a été soutenu par l'Institut de sciences ouvertes Tannenbaum de l'Institut neurologique de Montréal, le programme intégré en neurosciences de l'Université McGill, l'Institut universitaire de santé mentale Douglas, l'Association des étudiants au cycle supérieur en neurosciences et l'Association d'étudiants au cycle supérieur de l'Université McGill. Enfin, nous tenons à exprimer notre gratitude à ceux d'entre vous qui ont fait un don lors de leur inscription à l'événement. We greatly appreciate all of you attending and hope you learned something new today. To say thanks, we have arranged a raffle for all our registrants. The prizes include a $50 Uber Eats gift card and a $50 gift card for the Word bookstore. Special thanks to the Word for their generous donation. Nous apprécions grandement votre présence, et nous espérons que vous avez appris quelque chose de nouveau aujourd'hui. Pour vous remercier, nous avons organisé un tirage cadeau pour tous ceux qui ont participé à l'événement aujourd'hui. Les prix comprennent 2 cartes cadeaux de $50, l'une pour Uber Eats et l'autre pour la librairie The Word. Un grand merci à The Word pour ce don généreux. So the winner of the Uber Eats gift card is Delphine Brézif. Congratulations, you will be contacted shortly with additional details. La personne qui a gagné la carte cadeau de Uber Eats est Delphine Brézif. Félicitations, vous serez contacté sous peu avec des détails supplémentaires. So the winner of the Word gift card is Selma Hamdani. Congratulations, you will be contacted shortly with additional details. La personne qui a gagné la carte cadeau de Word est Selma Hamdani. Félicitations, vous serez contacté sous peu avec plus de détails. The presentations will be posted on YouTube in the coming weeks. Also, our upcoming event in the winter will be streamed on YouTube. So be sure to subscribe to the NeuroLingo channel and turn on notifications to receive updates. Please fill out the survey linked in the description to let us know how we did and how we can improve for the next event. Les présentations seront publiées sur YouTube dans les semaines à venir. De plus, notre prochain événement de l'hiver sera diffusé sur YouTube. N'oubliez donc pas de vous abonner sur la chaîne de NeuroLingo et d'activer vos notifications afin de recevoir des mises à jour. Veuillez également remplir le sondage qui est dans la description pour nous faire part de votre avis sur l'événement d'aujourd'hui et nous faire des commentaires sur comment on pourrait améliorer pour le prochain événement. Goodbye for now. Thank you. Stay safe and take care. Au revoir. Soyez prudents et prenez soin de vous.